these are lipids and these are proteins. So when carbohydrate is attached with the lipid directly, that is called glycolipid, and when it is attached with the protein, then it is called glycoprotein. See here. Actually, it is in the ring form I am representing here. And at the top, you might have studied
amylopectin. Some pictures were like this one.
Is here. This is carbohydrate chain. This is oligosaccharide. This is oligosaccharide, and this oligosaccharide is attached with the protein. Therefore, this whole structure is called glycoprotein. Glyco means carbohydrate, saccharide. So glycoprotein. Here it is attached with lipid directly. Therefore, it is called glycolipid. Here also it is attached with protein, therefore it is called glycoprotein. At the top of it, all these structures are negatively charged. That is due to presence of sialic acid. Actually, N-acetyl glucosamine is also called sialic acid. According to the greater membrane model of Lehinger, greater membrane model of cell membrane, this is the structure, and in that structure he has given the position of sialic acid, a component of many glycoproteins and glycolipids in animals. Understand? Now you can see here, so this is your N-acetyl neuraminic acid or sialic acid. Now reducing monosaccharides. Monosaccharides, when they are oxidized, if a molecule is oxidized, you might have heard oxidoreduction reaction. Whenever there is oxidation, another molecule is reduced, isn't it? So when we say reducing monosaccharides, it means monosaccharides will be oxidized. It means they are reducing agent. They are oxidized themselves and they reduce to others. Therefore, they are called reducing monosaccharides. Monosaccharides can be oxidized by cupric ion. Cupric ion oxidizes them, and therefore they reduce cupric ion into cuprous ion. The carbonyl carbon is oxidized to a carboxyl group. They are converted to aldonic acids, I described earlier. Second position, there is first position, there is change. This is the basis of principle of Falling's reaction and old diagnostic test for diabetes. You know, Falling's solution is kept in the urine, and then there is change in the color. Why? Because cupric ion is converted into cuprous ion. It is glucose is oxidized and cupric ion is reduced. Understand? So on the basis of that color change, it was diagnosed if there is sugar in the urine or no. But nowadays, you know, we do not use Falling's solution. Nowadays, kit has come. No? Even and the patient tests its own blood glucose level. You know it? What they do? They prick the needle and then put one drop of blood
On the
strip,
yes.
and then what they do? There is a holder type thing. No? When it is put in that, then it displays. What is that? That a small thing is what? It is a spectrophotometer. It is a spectrophotometer. What is the basis? Modern method is photometric measurement. What is it? D-glucose plus oxygen. That strip contains glucose oxidase. When glucose oxidase is present there, what will do? It will be, glucose will be oxidized because oxygen is there. It will be oxidized, and then there is formation of D-glucono-delta lactone plus hydrogen peroxide. And due to that hydrogen peroxide, whatever dye is present in that strip contains both things, that glucose oxidase and the dye. Glucose will be converted, and there is generation of lactone and hydrogen peroxide. That hydrogen peroxide will react with dye, and whatever color is generated, that is detected by photometer, and on that basis it gives you the result. What is the concentration of glucose in your blood? So nowadays patients also do. No? They buy a strip and that kit, they prick a needle, they take one drop of blood, they put on the strip, and they put in that a small, no, you can say, a pocket spectrophotometer. No? They put it in, wherever they go, they test it, what is the level of glucose in his blood. So, actually, always you ask, we should try to cut down the cost.
Yes.
No, it has been done, isn't
So
it?
now I was just thinking about the Felling's reaction and photometric. So is there a better way of doing it?
Of course. But what can be done in easy way without
Yes.
anything? No?
Yes.
We can.
Which dye?
Huh?
Which dye they
Dye, because it is again proprietary things. They do not disclose which dye is present in that strip. They do not disclose
And
it.
my doubt is that if it is he is a serious diabetic, once he pricks, will blood clot?
No, it takes time because they make very small. No, only one drop is required to measure it. So therefore, they do not damage a lot. No, only a small little is there. Now it is very important. Hemoglobin glycation, particularly the blood glucose level. Till now, what we did, we used to measure the glucose level in the plasma, but it varies. If you take food, its level is something else. If you are on fasting, the result will be something else. Sometimes you might have seen when you go for blood glucose test, so you have to come on without taking food, and when they take blood, after that they take glucose, and after half an hour or 1 hour, they take blood again. What is the effect of that glucose when it comes in the circulation? Then what is the level? Because normal level is 180 to 120, and when it is when food is taken at that time, maximum threshold level is 180 milligram per 100 milliliter. If it crosses that, even in the fasting time, at the fasting time, if blood is taken and it is crossing the 120, it means the person is diabetic. If food is taken and then it is crossing 180, then it shows the symptom of diabetes. But it is very confusing. Suppose you took food in the morning and you waited for some time and then blood was taken, or sometimes some people are their metabolic activities are very high, then it is confusing. Result is confusing. Therefore, now people are telling that this thing should be done. Hemoglobin glycation. If glucose binds with the hemoglobin, and if that thing is measured, then it will be accurate. It will give the accurate result. Why so? Tonight, transporters in electrocyte membrane equilibrates intercellular and plasma glucose concentrations. Actually, what happens? As I mentioned earlier, suppose this is your RBC. This is your RBC, and glucose transporter is present in it. This is your plasma. What level of glucose is present in the plasma? It enters. There is no any maintenance thing. It enters. And when it enters, this RBC, as you know, particularly erythrocyte, is filled with hemoglobin. When it is filled with hemoglobin, so that glucose binds with hemoglobin, and that process is called hemoglobin glycation. Generally, you might have heard glycosylation. Have you heard? Glycosylation. When people say protein glycosylation, so when protein is bound with carbohydrate or any type of glucose or lactose or whatever carbohydrate is there, if it binds with the protein in the presence of enzyme, that is called glycosylation, and it occurs in endoplasmic reticulum and Golgi complex. When there is synthesis of protein, after synthesis, particularly when protein is synthesized on attached ribosome, then that protein goes in the lumen of the endoplasmic reticulum. I describe it. Some people are seeing my face on it. As you know, this is your endoplasmic reticulum. This is your endoplasmic reticulum, and on the endoplasmic reticulum there is your ribosome, and when protein is synthesized, there is a channel-like structure in the larger subunit of ribosome. When this protein is synthesized, this nascent protein comes. After synthesis, it enters in the lumen of endoplasmic reticulum, and then there is post-translational modification. Translation means protein synthesis. When from this mRNA protein is synthesized, that is called translation. And when this protein enters in the lumen of endoplasmic reticulum, and there is certain change in the protein, that is called post-translational modification. So this post-translational modification occurs in the lumen of endoplasmic reticulum as well as in the lumen of cisternae of Golgi complex, cisternae of Golgi complex, and then that protein is packed in the vesicle and it is released. If it is membranous, then it goes in the membrane. If it is extracellular, then it is released outside. Okay? So that is whatever post-translational modification occurs here, particularly when protein is bound with carbohydrate, that occurs in the presence of enzyme that is called glycosylation. Here there is not any enzyme, there is simple binding of glucose with the hemoglobin in the absence of enzyme, therefore it is called glycation. Understood? Glycosylation and glycation difference you understood? Now, hemoglobin is directly in the contact of blood glucose concentration, as I mentioned. Directly transporters transport, there is no any concentration gradient. A non-enzymatic reaction occurs between glucose and the primary amino group in hemoglobin, either the amino terminal valine or the epsilon amino groups of lysine. As you know, when we say amino polypeptide chain here it is NH2 and here it is COH. This is amino terminal end and this is carboxy terminal end. Okay. So when hemoglobin valine is present at the amino terminal, okay, or when you arrange, when there's branched chain, side R group, particularly for amino acid, we write C R H NH2 COOH and R. This R is your alkyl or aryl. Alkyl or aryl chain. So if it is long chain, suppose C, then you have to consider alpha, beta, delta, gamma and epsilon. These are positions of that carbon. So on that basis, if in the lysine gamma amino groups, epsilon amino groups is, it will bind with the hemoglobin and then hemoglobin is called glycated hemoglobin. Okay? Glycated hemoglobin present at any time reflects the average blood glucose concentration. It does not tell what is the amount after taking food and what is the amount at the time of fasting. It tells average. So this is most appropriate. Glycated hemoglobin gives you the exact result, particularly what is the concentration of glucose in the blood. Understand? Last 2 weeks is the most important in setting the level of GHB, glycated hemoglobin. GHB is glycated hemoglobin. As you know, the lifespan of RBC is 120 days. In the last 2 weeks, it means old RBC. If old RBC are more, then it is more appropriate. Okay. Normal GHB values are about 5% of total hemoglobin. 5% of total hemoglobin. It means all hemoglobin will not be glycated. 5% only. And if it is 5%, then level of glucose will be 120 milligram per 100 milliliter. If 5% hemoglobin are glycated, it means blood glucose level is 120 milligram per 100 milliliter of blood. If it is 13%, then what will be? Particularly in the diabetic patient, it is around 13%. You can calculate it. Suppose a question is asked, glycated hemoglobin is this percent, then what will be the percent? No? You can answer it, isn't it? On the basis of percentage, you will tell that the person is hypoglycemic or normal or hyperglycemic. Understand?
So will there be error value?
Hmm?
Plus or minus 5%, something like that?
Of course, they will, if they will tell about hyperglycemic, always they will tell more than 10%. If they will tell hypoglycemic, less than 5%.
No sir, means we get, suppose we are calculating and we get around 110 mg per 100 ml.
110, okay.
So what do we consider? We
Okay,
tell
normal.
that normal.
Normal,
So
yeah.
that plus 10 minus 10 would be normal.
Yeah, of course, 80 to 120 milligram is normal. On that basis, you can say, suppose this type of question is asked. So if this type of question, they will give you the percentage of glycated hemoglobin, and then you will calculate what is the level of glucose.
So what is the measurement process of GHB? So like in the previous case, we were using photometry and all.
Mhm.
What is the process over here?
Of course, it will also react in the same way. No? It will react or by failing solution also. It will react. On that basis, you can calculate what is the percentage.
So how does a 5% correspond to this 120 mg only per 100 ml? Matlab it has been
No,
calculated.
actually it has been case to case. No? Actually it varies, but when research is done, no? When this is the percentage, then what is the blood glucose level? It is measured. No? At that time, it is that level. If it is 5%, the blood glucose level is 120 milligram, 120 milligram per 100 milliliter of blood.
So what is the normal, abnormal value? Matlab up to 10 it is normal, up to 7, up to
Generally 13, 13 and above hyperglycemia. Now effect of glycation. What happens when glycation occurs? Sometimes there is some harmful effect. What is the harmful effect? Glycation of hemoglobin involves rearrangement, oxidation and dehydration of carbohydrate moiety. It changes. Thus there is production of heterogeneous mixture of advanced glycation end products. When all these things happen, there is change in the molecule. And due to that change, advanced glycation end products can leave the erythrocyte and form the covalent bond with proteins. Actually, like this only. It happens in the case of plasmodium. So I described it. In the case of plasmodium, as you know, malaria is due to plasmodium. What happens when plasmodium enters inside our bodies, particularly when a female Anopheles mosquito bites? Why female Anopheles bites? Huh? Why not male?
live on fruit juice.
Huh? Plant juices, okay? Why not blood?
Because females lay eggs and they require blood.
For the egg laying by female mosquitoes, human blood protein is required. She's a real demon, isn't it? Because for egg laying, human blood protein is required. Without it, it cannot lay eggs.
It wants
use-
to give the best.
Huh? Yes.
So therefore all
For
mosquitoes are not Anopheles. There are some Anopheles. That's why-
Anopheles, Culex, Aedes. No?
That's why there are
There-
varieties.
Varieties. Culex fatigans, isn't it? Therefore, there are two types of concept. Some mosquitoes are active in evening and morning, and some are active in night.
This female Anopheles, when it is active.
Actually, on female, I'm not sure at what time it is. Actually, I read it, but I forgot at what time exactly. Particularly Culex. Culex remains active at night. Therefore, Anopheles in the morning and evening. Anopheles. Because Culex spreads Phalidia. That what? Phalidia vermin. Wuchereria bancrofti. Wuchereria bancrofti is transmitted by Culex. And Plasmodium is transmitted by Anopheles. Therefore, generally, the test of Phalidia, when in daytime blood is taken, it does not show positive. Because at the time of when human remains active, at that time it goes in the deep circulation. And when humans become, you can say in the resting stage, then it comes in the peripheral circulation. Particularly at night, human sleep, and at that time, if it is active only, then the Culex will get the macrophallaria. Only then it will transmit from one individual to another individual. Therefore, Culex remains active at night. That is.
So even for malaria, the hemozoin is produced at night. For malaria, the hemozoin is produced at night. So
What told you?
people suffer for
What told you? Who told you it is processed like that?
Not,
No, actually there are 4 types of meridian. Plasmodium vivax, Plasmodium falciparum, Plasmodium mallory, and Plasmodium hobble. So they are on different, cycle is different, 48 hour, 36 hour, 48 hour, 72 hour mixed. At different time periods they complete their life cycle. And when life cycle is completed, at that time they release a large, particularly erythrocytic cycle is completed in different time periods. On that basis they release hemozoin. Okay. So in the case, what happens when Plasmodium takes hemoglobin? The hemoglobin is eaten by Plasmodium. As you know, hemoglobin contains 2 parts of part. Metallic part is cytochrome, and the proteinous part is globin. So that globin-based protein, that proteinous part is eaten by the parasite, or you can say parasite utilizes the proteinous part for its metabolic activities, and excretory material is discarded, that is Sucrose dot in the case of Plasmodium. And that heme part, it is unable to metabolize the heme part. If heme remains inside the RBC or inside the Plasmodium, suppose this is your Plasmodium, and it has heme, it takes hemoglobin, and heme part is in monomer form. In single units it is present, then it is cytotoxic. Monomer of monomeric form of heme is cytotoxic. Therefore, the parasite will die. But it does not die. Why not? Because it has a special gene, and that synthesizes an enzyme. That enzyme polymerizes the heme. And when heme is polymerized, all heme is connected. Now it becomes hemojoin. That heme, monomeric form of heme, is not harmful for human, but it is harmful for parasite. To protect the parasite, nature has given a gene. So it polymerizes hemojoin, it polymerizes heme, then hemojoin forms. Hemojoin is harmless for the parasite, but poisonous for human, toxic for human. When the erythrocytic cycle completes, at that time there is bursting of RBC and it is released in the blood. When that level goes high, at that time there is fever. Okay, to release of that. Understand? So like that, when there is a glycation product in product forms, it is released by the RBC, or it can come out from RBC. And when it comes out from the RBC, then what happens? Covalent bond, it forms covalent bond with other proteins. Whatever proteins are present in different tissues, and particularly thus it damages kidney, retina and cardiovascular system. Diabetes people sometimes they become blind. You might have heard vision decreases, no? Kidney, sometimes kidney failure due to that. Like I said, when there is more amount of glucose, it will go to the RBC, and more amount of glycated hemoglobin will form. Then there is more production of glycation in products, and then it will be released in the blood in the more amount, and then it will bind with different types of proteins in different tissues, and it will damage these body parts.
So, one question is, when you are referring to heme, you mean the porphyrin ring?
Yeah, the porphyrin.
It will polymerize the porphyrin ring?
Yeah, porphyrin ring. That is hemojoin. That's all. Okay. Actually, till now we finished only monosaccharide. Nothing else. Monosaccharide.